Quantitative 2D color image analysis is a powerful technique to extract information from microscopy images. As an example, we create a new workflow. We can start simple by selecting the files to process and using the Import Image task. Ensure that the Convert RGB to Grayscale option is set to No to preserve the color information for the analysis. Similarly, confirm that Convert 2D Stack to 3D is set to No when processing individual 2D images. If the image format metadata does not contain information about the image resolution, manually override and set the correct resolution. This step is critical for accurate quantitative measurements involving distances and areas. Now we are ready to start the workflow. It is advisable to select a few files for initial processing to test and refine the analysis parameters. We can now dynamically extend the workflow step by step to quantify different properties. For this purpose, we repeatedly append different tasks. For example, the first step might be appending a Gaussian filter task to the workflow to reduce noise. Previewing or comparing any result or intermediate images is easily possible at any point in the workflow. Next, we append the interactive color thresholding task. Here, a red overlay indicates the objects identified based on the defined color range. Different color spaces can be used. Here, the hue and saturation range sliders are used to select the features of interest. In this example, we measure the blue-stained cells. If necessary, the current job can be restarted to further adjust and fine-tune the color thresholds. Once satisfactory settings are found, restarting all runs can apply these consistent parameters across the entire dataset. Now, we have a simple binary mask, and many tasks are available to work with these, like morphological opening or closing operations that bridge or separate small gaps or holes within the segmented object mask. Adjust the closing distance parameter to effectively close larger holes as needed. Similarly, component labeling can filter out ranges of objects based on their size, effectively removing small, irrelevant artifacts from the analysis. We now append a basic 2D labeling measurement task to quantify various properties of each individual segmented object, including its area, perimeter, and roundness. Many other parameters can be quantified, like the local thickness at each pixel within the segmented objects, providing further information about their structure and distribution. For rigorous quantitative analysis of these color images, the goal might be to accurately count cells or grains, to measure tissue area and density, and analyze particle distributions with respect to their size, shape, and orientation. There are many tasks available to segment, count, measure, and analyze all kinds of features and distributions. Pre-trained machine learning models can easily be applied, and new models can be trained using existing data, or new annotations can be prepared. Review the initial results and fine-tune the parameters of each processing step as necessary to optimize the segmentation and measurement accuracy. Generate a table report to summarize the quantitative measurements across all processed images. Once the workflow works for the initial dataset, expand the analysis by adding more image runs. The previously defined workflow and parameters will be automatically applied to these new runs. The View Sequence feature gives an overview of all processed runs so far. You can navigate to examine the results of a specific run in detail. Snapshot tasks allow visually displaying and comparing all data in a consistent way. For example, the original image can be compared to the generated local thickness map, overlaying them with transparency automatically for all runs. Now we further extend the analysis by adding the full set of over 240 images. Observe how the new results populate the sequence view over time. The table report also refreshes to include the data from these new runs. The individual per object measurements can be viewed or exported as a table where each row contains all the measured parameters of one particular structure. For faster processing on powerful computers, the worker can be configured to process multiple runs concurrently, such as 10 at a time. Problems can be spotted easily. The visual overview allows for easy identification of potential issues. One option is to adjust the thresholding parameters specifically for the problematic runs only. To obtain the results for this specific run quickly, its priority can be set to high. The Snapshots Report feature allows reviewing the generated snapshots, together with the favorited starred parameters and quantified values. The Chart Report feature allows quickly comparing different measured parameters across the analyzed objects and runs. Correlate these charts with the corresponding snapshots to visually investigate any identified outliers or trends. Similar workflows can be used, for example, for histopathology studies, histomorphometry of histological sections, with staining agents that selectively color different cell types or components, but also, for example, for petrographic thin sections, and more.